different arms of tRNA, students? First arm, acceptor arm. What's the function of the acceptor arm? Binds to amino acid. The second arm is known as T-zy-C arm or pseudo-uridine arm. It binds to ribosome. The third arm, yes, anticodon arm. It binds to mRNA. The next arm, DHU arm, dihydrouridine arm, binds to amino acid synthetase enzyme. As a whole, what is the function of tRNA? Now you have seen the function of each arm, is it not? As a whole, what is the function of tRNA? Its function is to transfer amino acid from the cytoplasm to the ribosome for protein synthesis, right? It carries what? Amino acid. The last type of RNA. Which is the last type of RNA, students? Ribosomal RNA. Idra concentration mele, idra arms mele, questions have been asked. Right? So whatever you all don't know, make a note of it. Right? rRNA. Ribosomal RNA. Yes. Now ribosomal RNA, rRNA. This is the largest, smallest yaudu? tRNA. Short-lived yaudu? Ide kelidare. Short-lived yaudu? mRNA. Largest yaudu? rRNA. And maximum ನಮ್ಮ cytoplasm ಅಲ್ಲಿ ಇರೋದು ಯಾವುದು? rRNA. Yes. So this, its concentration in the cytoplasm is around 7. Which is the most abundant RNA? This is asked. rRNA, right? Which is the shortest RNA? tRNA, right? Now what is the function? We are not studying the structure of rRNA. Directly we are going to study the function of rRNA. One function is it helps in maintaining the structure of ribosome. rRNA, ribosomal RNA. It helps in, yes, maintaining the structure of ribosome. And this question is frequently asked. There is an evidence. There are different types of rRNA. 16S rRNA, 23S rRNA, 5S rRNA anta bere taradu rRNA is present. And there is evidence that 23S rRNA untalla, 23S rRNA, this functions as one enzyme. ಇದು ಒಂದು enzyme ತರ function ಮಾಡ್ತದೆ, and the name of this enzyme is ribozyme, not ribosome. Ribosome is an organelle. So 23S rRNA functions as an enzyme, ribozyme. Now what is the function of this enzyme? This enzyme helps in the formation of, what is this? ಅಮೈನೋ ಆಸಿಡ್. ಅಮೈನೋ ಆಸಿಡ್ಗಳ ನಡುವೆ ಇರುವ ಬಾಂಡ್ ಅನ್ನ ಎಂತ ಅಂತ ಹೇಳೋದು? ಎರಡು ಅಮೈನೋ ಆಸಿಡ್ಗಳ ನಡುವೆ ಇರುವ ಬಾಂಡ್ ಅನ್ನ ಎಂತ ಅಂತ ಹೇಳೋದು? ಅಲ್ಲ, ಅದು ಶುಗರ್ ನಡುವೆ ಇರುವ ಬಾಂಡ್ ಅನ್ನ ಗ್ಲೈಕೋಸಿಲಿಕ್ ಬಾಂಡ್ ಅಂತ ಹೇಳೋದು. ಪೆಪ್ಟೈಡ್ ಬಾಂಡ್, ರೈಟ್? ದಿಸ್ ಇಸ್ ಪೆಪ್ಟೈಡ್ ಬಾಂಡ್. So what's the function of ribozyme? It helps in the formation of peptide bond between amino acid during protein synthesis, right? It helps in the formation of peptide bond between amino acid during protein synthesis, right? Yes. So these are the different types of what? rRNA. So which is your next question, students? What is that in the match the following? Okay. I don't have that. Okay. Yes. So they have mixed the questions, that's all. Quick, can you match it, students? Translation. Translation is what? Yes? What is translation? Is translation ಅಲ್ಲಿ ಯಾವ enzyme involve ಆಗಿರೋದು? Amino acyl tRNA synthetase. Translation process ಓದಿರೋದು ನೆನಪಿಲ್ವಾ? Which is the enzyme involved there? Amino acyl tRNA synthetase enzyme. Do you know the function of this enzyme? Translation ಅಲ್ಲಿ first step ಏನು? What's the first step under translation, students? Activation. Correct? First step is activation and selection of amino acid. First step is activation and selection of amino acid. Amino acid illirodu cytoplasm alli. These amino acids are not active. They are inactive. They have to be activated. Translation alli ಇವುಗಳು participate ಮಾಡಬೇಕು ಅಂತ ಇದ್ರೆ amino acid has to be activated, right? So ಯಾರು activate ಮಾಡೋದು ಈ amino acid ಅನ್ನ? Amino acid is inactive, right? This is inactive. It is activated by ATP and enzyme. And the enzyme that is involved here is amino acyl tRNA synthetase. Right? Now this activates the amino acid, and the activated amino acid is called amino acyl AMP. ATP is becoming AMP enzyme. Enzyme also binds. Plus 2 inorganic phosphate. Yeredu inorganic. ATP is becoming AMP. Adenosine triphosphate is becoming adenosine monophosphate. Right? Yes. So, amino acid ivaga activate aitu. Next ಏನಾಗ್ತದೆ? Amino acid activate aitu. ಆನಂತರ ಏನಾಗ್ತದೆ? Now this activated amino acid reacts with the tRNA. Amino acyl AMP enzyme reacts with which one, students? tRNA. Right? And this process is known as charging. Idanna charging anta helodu. Right? Yes. Now, ಈ activated amino acid tRNA ಗೆ ಹೋಗಿ bind ಆಗ್ತದೆ. So, this is the tRNA, right? We have drawn this tRNA there. This is the 5' end, 3' end. ಎಲ್ಲಿಗೆ ಹೋಗಿ ಅಮೈನೋ ಆಸಿಡ್ ಬೈಂಡ್ ಆಗ್ತದೆ? 3' ಎಂಡ್ಗೆ. ಸೊ ಈ ಆಕ್ಟಿವೇಟೆಡ್ ಅಮೈನೋ ಆಸಿಡ್ ಇಲ್ಲಿಗೆ ಬಂದು ಬೈಂಡ್ ಆಯ್ತು. Right? And this is known as amino acyl tRNA. ಇದನ್ನ ಎಂತ ಅಂತ ಹೇಳೋದು? ಅಮೈನೋ acyl tRNA ಅಂತ ಹೇಳೋದು. And what is got? Plus AMP plus enzyme. AMP ಮತ್ತೆ enzyme is got back. Right? Okay, so translation ige, what's the option there? Amino acyl tRNA synthesis. What's the function of this enzyme, students? Activation of amino acid, right? Transcription ige, RNA polymerase, right? And DNA replication ige, Okazaki fragments. What are Okazaki fragments? The short fragments that are synthesized. How are these fragments joined? With the help of which enzyme? DNA ligase. Right. So it is option B. Right? Yes. Okay. So the next question. Select the incorrect matched pair. Incorrectly matched pair. Initiation AUG. GUG. AUG can rarely become GUG. Okay. Stop codons or nonsense codons. UAG, UAA, UGA all begin with uracil. Right. Methionine. AUG codes for methionine. Anticodon. ಎಲ್ಲಿರಬೇಕು ಆಂಟಿಕೋಡಾನ್? tRNA ಅಲ್ಲಿ. ಆದರೆ ಇಲ್ಲಿ ಏನು ಕೊಟ್ಟಿದ್ದಾರೆ? mRNA ಅಂತ ಕೊಟ್ಟಿದ್ದಾರೆ. So it is incorrectly matched. Yes. So our next question. During transcription, the site of DNA molecule at which RNA polymerase binds. I think we ಎಲ್ಲಿಗೆ RNA polymerase bind ಆಗೋದು? Promoter gene ಗೆ. Yes. Promoter gene ಗೆ. Yes. Next question. What would happen if a gene coding a polypeptide of 50 amino acid? 50 amino acids ಗಳು ಉಂಟು ಅದರಲ್ಲಿ. Right? ಆದರೆ 25th codon ಯಾವುದು? UAU ಇತ್ತು, ಮ್ಯೂಟೇಟ್ ಆಯಿತು. ಏನಾಯಿತು? UAA ಆಯಿತು. 25th codon. UAA is what? Stop codon. So 25th amino acid synthesize ಆಗ್ತದಾ? ಇಲ್ಲ. ಯಾಕಂದ್ರೆ ಅಲ್ಲಿ ಪ್ರೋಟೀನ್ ಸಿಂಥಸಿಸ್ ಸ್ಟಾಪ್ ಆಯಿತು. ಸೊ ಎಷ್ಟು ಅಮೈನೋ ಆಸಿಡ್ಗಳು ಸಿಂಥಸೈಸ್ ಆಯಿತು? A polypeptide of 24 amino acid will be formed. Right? Yes. Okay. Is it match the following? No, ma'am. Both deoxyribose and ribose belong to a class of sugar called? Yes, because it has got 5 carbon atoms. What's the molecular formula of ribose sugar, students? C5H10O5. Ribose is O5. Deoxyribose is O4. Right? Match the following. Okay. Can you match it? Frederick Mixture is nuclein. He called DNA as nuclein. Griffith worked on Streptococcus pneumoniae. Hershey and Chase, they worked on bacteriophages. Watson and Crick, DNA double helix. Wilkinson, Franklin. Right? Yes. So once you are thorough with the synopsis, once you are thorough with your, concepts, solving question will not take much time, right? What's our next question? Dash was the first genetic material. No. RNA. RNA was unstable. That is why DNA evolved. Evolution ಓದಿದಿರಲ್ಲ, I think our next topic itself is evolution. So evolution ಅಲ್ಲಿ evolve ಆಗಿರುವಂತ first genetic material ಯಾವುದು? It is RNA. RNA unstable ಯಾಕೆ? RNA is unstable. Why? Yes, students, why is RNA unstable? We have written the structure of deoxyribose, correct? First carbon atom, second, third, fourth, fifth. What is present here? OH, H, H and H, H and OH, CH2, OH, H. This is deoxyribose, right? RNA, ribose sugar, first carbon atom, second, third, fourth, fifth. Same OH, H, H, OH. Here it is H, here it is OH. HOH, rest of the things same. CH2OH, H. What is more in ribose sugar? OH group. So that is the reason why ribose sugar of RNA is highly unstable and highly reactive. Unstable ಮತ್ತೆ reactive. ಅದೇ DNA ಅಲ್ಲಿ ಯಾವ OH ಗ್ರೂಪ್ ಕೂಡ ಫ್ರೀ ಇಲ್ಲ. ಇಲ್ಲಿ ಎಂತ ಉಂಟು? ನೈಟ್ರೋಜಿನಸ್ ಬೇಸ್. ಇಲ್ಲಿ ಎಂತ ಉಂಟು? ಫಾಸ್ಫಾರಿಕ್ ಆಸಿಡ್. And what is attached here? ನೆಕ್ಸ್ಟ್ ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್. ನೆಕ್ಸ್ಟ್ ಇದು ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ ಅಟ್ಯಾಚ್ ಆಗ್ತದೆ. ಸೊ ಯಾವ OH ಗ್ರೂಪ್ ಕೂಡ ಇಲ್ಲಿ ಫ್ರೀ ಇಲ್ಲ. ಅದೇ RNA ಅಲ್ಲಿ ಇಲ್ಲಿ ನೈಟ್ರೋಜಿನಸ್ ಬೇಸ್, ಇಲ್ಲಿ ಫಾಸ್ಫೇಟ್, ಇಲ್ಲಿ ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್. ಒಂದು OH group is free. That is the reason why RNA is unstable and highly reactive. Right? Okay. So which is our next question? If the sequence of bases in DNA the bases anna kottidare, then the sequence of bases in its transcript ಅಂದ್ರೆ RNA, ಟ್ರಾನ್ಸ್ಕ್ರಿಪ್ಟ್ ಅಂದ್ರೆ ಏನು? RNA. ಕಾಂಪ್ಲಿಮೆಂಟರಿ ಅಂತ ಕೇಳಿದ್ರೆ DNA ಅನ್ನ ನೀವು ಫೈಂಡ್ ಮಾಡಬೇಕು. Now you have to find the transcript, that is RNA ಅನ್ನ ಫೈಂಡ್ ಮಾಡಬೇಕು. Can you do it? Try doing it. Guanine pairs with cytosine. Cytosine with guanine. Yes. Thymine with adenine. Option C. Fourth option. Right. Sorry, third option. Not fourth, third one. Right? Next question. Which out of the following statement is incorrect anta kelidare? And this question was very, very important. Right? Features of genetic code. Option A. Genetic code is non-ambiguous, right? Athava unambiguous. All the others are correct alvalli. Non-overlapping, degenerate, universal. What is degenerate, students? ಒಂದು ಅಮೈನೋ ಆಸಿಡ್ಗೆ ಒಂದಕ್ಕಿಂತ ಜಾಸ್ತಿ ಕೋಡಾನ್ಸ್ಗಳು ಉಂಟು ಅಂತ. ನಾನ್ ಆಂಬಿಗ್ಯುವಸ್ ಅಂದ್ರೆ ದೇ ಆರ್ ಹೈಲಿ ಸ್ಪೆಸಿಫಿಕ್. AUG ಎಂತದನ್ನ ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡ್ತದೆ? ಮಿಥಿಯಾನಿನ್. and throughout the life it produces only methionine. So it is highly specific. Okay. So which is our next question?
Refer the given sequence of steps and select the correct option.
Refer the given
Sequence of steps.
which one?
20.
Okay, yes. Refer the given sequence of steps and select. This we have written, is it not? Central dogma this is. So can you identify the right option there? Right. DNA synthesizing heterogeneous nuclear RNA is transcription. RNA synthesizing mRNA is splicing. Right. And mRNA synthesizing protein is right. Translation. Yes. So next question. Methyl guanosine triphosphate is added to the 5' end of hnRNA. What is this process called? Right, capping. If the sequence of bases in one strand of DNA is given, what would be the sequence of base on complementary strand? This means you have to find the sequence of DNA, RNA alla, complementary strand kelirodu. Option C. Match madidre, so that becomes your option. Yes. Next, what do you have? Match the following. Which of the following steps of transcription in prokaryotes is catalyzed by RNA polymerase? I mentioned this. All of these. Initiation is catalyzed by RNA polymerase. Elongation is catalyzed by RNA polymerase. Termination is also catalyzed by RNA polymerase. But factors help. Initiation ಗೆ ಯಾವ factor help ಮಾಡೋದು? Sigma factor. Termination ಗೆ ಯಾವ factor help ಮಾಡೋದು? Rho factor. Right? Yes. Now you all have the match the following there? No. Okay. Yes. What's your next question? Which of the structural gene? Okay. The structural gene in eukaryotes possess coding ಮತ್ತೆ non-coding region. ಇದನ್ನ ಎಂತ ಅಂತ ಹೇಳೋದು? Coding region ಅನ್ನ exon ಅಂತ ಹೇಳೋದು. Non-coding region is known. Right. Introns. Right? Yes. The next question. If the sequence of bases in coding strand of DNA they have given, then the sequence of base in mRNA. Let's see who will do it. Option there. Listen. Coding strand alli ಕೊಟ್ಟಿರೋದು. ಕರೆಕ್ಟಾ? See, sometimes to confuse you all, ಈ ತರದ question ಕೊಡ್ತಾರೆ. Coding strand alli ಕೊಟ್ಟಿರೋದು. Is the coding strand involved in the synthesis of RNA? No. Which strand is involved in the synthesis of RNA? Template strand. So you have to find the template strand. Template strand ಅನ್ನ find ಮಾಡಬೇಕು. So template strand ಇದು ಹೇಗೆ find out ಮಾಡ್ತೀರಾ? ಏನು ಕೊಟ್ಟಿದ್ದಾರಲ್ಲಿ? ಕೋಡಿಂಗ್ ಸ್ಟ್ರಾಂಡ್ ಅಲ್ಲಿ ಎಂತದನ್ನ ಕೊಟ್ಟಿದ್ದಾರೆ? ಕೋಡಿಂಗ್ ಸ್ಟ್ರಾಂಡ್ ಅಲ್ಲಿ they have given A, T, T, C, G, A. This is coding strand. Coding strand, is it involved in the synthesis of RNA? No. That means you have to find the template strand. Adenine pairs with thymine, thymine with adenine, adenine, cytosine, thymine, adenine. Now you got the template strand. ಈಗ ಟೆಂಪ್ಲೇಟ್ ಸ್ಟ್ರಾಂಡ್ ಇದೆ, ಎಂತದನ್ನ ಫೈಂಡ್ ಔಟ್ ಮಾಡಬೇಕು ನೀವು? mRNA. So this is A pairs with T pairs with adenine, A with U, A with U, G with C, C with G, T with A with U. Idea the option?
Yes, ma'am.
Yes, so that's the option. Remember students, coding strand alli kotre first template strand anna find out ಮಾಡಿ ಆ ನಂತರ RNA ಅನ್ನ find ಮಾಡಿ. Yes. Okay, next match it. Yes, can you all match it? Try matching it. Option. Why is it there are so many options B, C? Option 2, B. Option 2, B. Is it right? Yes, moving on to the next question. Polycistronic messenger RNA usually occurs in bacteria entadu. So it is both A and B. Bacteria is also prokaryote and prokaryote is also the right one. So both A and B. The mutation that involves addition, deletion or substitution of single gene. Yes, ಒಂದೇ nitrogen base change ಆಗೋದು. Point mutation. Point mutation ಗೆ ಒಂದು example ಯಾವುದು? Sickle cell anemia. Right? GAG becomes GUG. Right. Sickle cell, next question itself is that one. Sickle cell anemia results from a single base substitution in a gene. Thus, it is an example of point mutation. Silent mutation andre entadu? Silent mutation. You know, students, mutation do happen in our body. It happens when you expose your skin to radiations, right? It may be the radiations that are used in X-rays, MRI, CT scan. ಈ ರೇಡಿಯೇಷನ್ಸ್ಗೆ ಎಕ್ಸ್ಪೋಸ್ ಮಾಡಿದಾಗ ಕೂಡ ಮ್ಯೂಟೇಷನ್ಸ್ ಆಗ್ತದೆ, ಅಥವಾ ನಾವು ನಮ್ಮ ಸ್ಕಿನ್ನನ್ನು ಸನ್ಲೈಟಿಂದ ಬರುವಂಥ ರೇಡಿಯೇಷನ್ಸ್ಗೆ ಎಕ್ಸ್ಪೋಸ್ ಮಾಡಿದಾಗ ಕೂಡ ಮ್ಯೂಟೇಷನ್ಸ್ ಡೂ ಹ್ಯಾಪನ್. ಬಟ್ ಯು ನೋ ಸಮ್ಟೈಮ್ಸ್ our body does not show any change. Mutation ಆಗಿರ್ತದೆ, ಆದರೆ ಆ mutation does not harm our body, right? It is not affecting our body. ಇಂಥ ಮ್ಯೂಟೇಷನ್ಸ್ಗಳನ್ನು ಸೈಲೆಂಟ್ ಮ್ಯೂಟೇಷನ್ಸ್ ಅಂತ ಹೇಳೋದು. It happens, but you know it does not affect much. ಯಾಕಂದರೆ ಇಲ್ಲಿ ಒಂದು ವೋಬಲ್ ಹೈಪೋಥಸಿಸ್ ಅಂತ ಇದೆ. ಒಂದು ನೈಟ್ರೋಜಿನಸ್ ಬೇಸ್, ಥರ್ಡ್ ನೈಟ್ರೋಜಿನಸ್ ಬೇಸ್ ಚೇಂಜ್ ಆದರೆ you know it does not affect much. Okay, which is our next question?
The fact that these purine base always stays through hydrogen bonds in the
Okay, the fact that a purine base always pairs through hydrogen bond with a pyrimidine base leads to dash in the DNA double helix, uniform width throughout DNA, right? Uniform width throughout. Yes. Synthesis of DNA from RNA is explained by all of this. I gave you that central dogma, then I told you reverse transcription, I mentioned about feminism also. So it is all of these. Yes. So what's our next question? Match the following. Yes, try matching it, students. Whatever you know, you match first and then look into the option. Option 2, Alec Jeffrey ಏನನ್ನ ಕಂಡು ಹಿಡ್ದವರು? DNA fingerprinting. F. Sanger, automated DNA DNA sequence ಎಲ್ಲಿ ಯೂಸ್ ಮಾಡ್ತೀವಿ ಆಟೋಮೇಟೆಡ್ DNA sequencing ಅನ್ನ ಹ್ಯೂಮನ್ ಜೀನೋಮ್ ಪ್ರಾಜೆಕ್ಟ್ ಅಲ್ಲಿ ರೈಟ್ ಎಕ್ಸ್ಪ್ರೆಸ್ ಸೀಕ್ವೆನ್ಸ್ ಟ್ಯಾಗ್
lack of
ಅಲ್ಲ ಜೇಕಬ್ ಅಂಡ್ ಮೋನಾಡ್ ಅಲ್ಲಿ ಐ ಜಸ್ಟ್ ಈ ಎಕ್ಸ್ಪ್ರೆಸ್ಡ್ ಸೀಕ್ವೆನ್ಸ್ ಟ್ಯಾಗ್ EST ಹ್ಯೂಮನ್ ಜೀನೋಮ್ ಪ್ರಾಜೆಕ್ಟ್ ಅಲ್ಲಿ ಯೂಸ್ ಮಾಡ್ತೇವೆ ಅಲ್ವಾ Every macleoid McCarthy glycine. Next question. The three codons which results in the termination of polypeptide chain synthesis. ಯಾವುದು ಮೂರು codons stop codons ಗಳು ಯಾವುದೆಲ್ಲ UAA, UAG, UGA all begin with uracil. Option 2. Yes. Next one. In an animal cell, the process of transcription and translation. Animal cell alli transcription elli agodu nucleus alli. Translation elli agodu cytoplasm alli. Ade prokaryotes alli transcription ಮತ್ತೆ translation ಎರಡು ಕೂಡ ಎಲ್ಲಿ ಆಗೋದು ನ್ಯೂಕ್ಲಿಯಸ್ ಇಲ್ಲ ಅಲ್ವಾ ನ್ಯೂಕ್ಲಿಯರ್ ಮೆಂಬ್ರೇನ್ ಅಂತ ಅಲ್ಲಿ ಎಂತದೂ ಇಲ್ಲ so it is in the cytoplasm right during translation is that the question yes, during translation activated amino acids get linked to tRNA this process is commonly called as charging amino acylation of tRNA both A and C right yes okay match it so for this you need to know those 64 codons so this is not a CET question because under CET this is not asked it's asked under NEET 64 codons and these 61 codons are sensible codons and the remaining 3 are nonsense codons yes so it is UUU is phenylalanine GGG is glycine UCU is serine CCC is proline and AUG is methionine option A yes so I think it's almost time alva so yes students so remaining questions you all can try and if you all have any doubts you all can ask right